Hello everyone. What I'm going to talk about in this lecture is the theory behind the polymerase chain reaction. So we already talked about that in biochemistry, but I'm going to expand on some aspect of PCR, particularly focusing on the topics of primer design, you'll see later on, and the application, the expansion in terms of usage of PCR to different applications and different techniques developed from PCR. And then I'll finish up touching a little bit about agarose gel electrophoresis, the reason being that you'll be using this in the lab in the next week. So these are the topics we're gonna touch here. As you can see, the main focus is on PCR. So first we'll go through an overview of PCR. We'll see how it works. And then we'll talk about the role, the importance, the key role of the primers, which are the those sequences of DNA that ultimately make PCR specific or able to replicate specifically a sequence within a DNA template. Then we'll talk about application of PCR, particularly focus on an example, how it can be used in forensic studies to investigate what they're called variable number of tandem repeats. Then we'll talk about how PCR is being modified through times, focusing particularly on one technique, which is quantitative real-time PCR, which is used pretty much anywhere now in all labs. I'm pretty sure that the day you start working, most likely you will run into that or you have to perform that. So we'll touch on the theory of quantitative real-time PCR as well. And then in the end, I'll just finish up refreshing some concept about agarose gel electrophoresis, how it works and how it can be used to visualize DNA molecules and to estimate their size. So let's start with an overview of PCR. So this is a technique that's been around for about 40 years, was invented first by this scientist here, Kerry Mullis, who made millions of dollars out of that by patenting it. And ultimately, what this technique is, if you think about it, is a simplified version of DNA replication. If you want to understand how it works, always put it in the context of DNA replication. It's a simplified version, very simplified, where everything has been streamlined so that you can use the minimum amount of reagents, ultimately, to replicate not a full genome, like DNA replication would do, but a very specific sequence, what we call the target sequence, the one you would see here in right here indicated as target. So that's a technique that allows you to do DNA replication but of a specific target right there. Okay, now how is this achieved? It's achieved, as I said before, by trying to minimize the number of reagents that are needed. So what do you need to do a PCR? Well, you need to mix very few components. Well, obviously you would need a template DNA. That's that DNA sequence that contains the target that you want to amplify. Now you can direct the DNA polymerization to the specific target. If you remember, in DNA replication, DNA polymerase is started, the polymerization from DNA polymerase started always by RNA primers made by primates. In this case, you don't want to add more enzymes. So what you want to do is replace the RNA primers with DNA primers that don't need to be made over and over by an enzyme. Not only that, but you can actually use these two primers to direct where the DNA polymerase has to copy the DNA and therefore direct it towards the target sequence. Okay, we'll talk about the design. That's what I'm going to focus mostly on later on, the design of the primers. These are the key element in PCR. Together with that, obviously you will need the DNA polymers. Usually is a TAC or a modified version of a TAC DNA polymers. Now there are other versions too. And obviously you will need also all the deoxyribonucleotides, so the components that are polymerized by DNA polymers, to copy the target sequence. Now obviously, together with that, since you're using an enzyme, TAC DNA polymerase or another polymerase DNA polymerase type, you would need some sort of buffer that would optimize the salt concentration and the pH to maximize the activity of this enzyme. But as you can see, this is a simplified version of DNA replication. However, designed to copy a specific target, minimizing the number of reagents that are necessary to do them. Now, how do you achieve the amplification, as we say, so making millions, billions of copies of a specific sequence, is by doing three specific steps repeated several times, which what referred to as cycles. So PCR consists of several cycles in the order of 30 to 35. Can go a little bit lower, so it depends on your application. But each of these cycles will consist of these three steps. The first one called denaturation, second one annealing, and the third one elongation. Let's put them again in the context of DNA replication. Denaturation is nothing else than the equivalent of the first step in DNA replication, which is the unwinding of the DNA. Some people call it the initiation of DNA replication. If you remember, in DNA replication from Bio 2, in that case you have this enzyme called helicase that opens up, then you have the single strand binding protein involved to keep them open, and so on, topoisomers. In this case, everything's simplified. You don't want to use additional enzymes and so on. All you use is temperature. So the first step in each cycle is what is called denaturation, is done at 95 degrees and achieves exactly the same thing that you achieve in DNA replication by opening up, separating the two strands of DNA. The second step is annealing. Annealing is that key step that ultimately directs later on the DNA polymers to the sequence you want to copy. And is that step done in temperature variable, we'll talk about that later on, how you choose them, between 50 and 65 degrees Celsius. And that's a step where the primers that you have designed will bind beside the target sequence. So this will be the target sequence that you're trying to make several copies of. And now the primers will be designed to bind right beside it. Okay, so these are designed to be complementary ultimately to the sequence in the opposite DNA, single-strand DNA molecule. Once this is achieved, then each cycle moves to the third step, which is elongation. This is done at a higher temperature than the annealing, so between 68 to 72 degrees Celsius. And this is when actually the DNA polymers now will start copying or expanding the primers, starting from the 3', so going 5' to 3', will start copying the complementary sequence in the target sequence in the template DNA. As you can see, so this is repeated. So after elongation, you will get a second cycle going back to denaturation, annealing, elongation, and so on. This will be repeated over and over for 30-35 cycles. So what happens here? Now you have that in each cycle, theoretically, every molecules get duplicated. You can visualize it here. So imagine this being your first cycle. This is your template DNA with a target sequence here, which is the blue sequence here. So in the first step, you would have your denaturation, annealing, so the primers would then bind to the target sequence there, and then elongation. So at the end of the first cycle, you would have had that the first copy would have been duplicated into two different copies. Now, if you move on to a second cycle, so you go back to denaturation and then annealing again and then elongation again, now you would have that each of these two copies would open up and would act as a template for the primers to bind to, being elongated, and therefore you would end up having that these two copies would have become 4 of them. Now if you keep going, third cycle, the same thing would happen. What I want to point to you here is two things. First of all, how in the long run, what you're really gonna be amplifying, at the beginning you have that the DNA polymerase goes past the target sequence. You can see here and here as well. This at the beginning is predominant in your sample, but in the long run, what you're really gonna be amplified is what we refer to as the amplicon, which is the sequence between the beginning and the end of the two primers. The beginning of what is called the left primer and the end of the right primer. We'll talk about that later on. So this piece here, the amplicon, is the one that will be later on becoming the template and becoming predominant at the end of your 30-35 cycles. And that's what you would see ultimately in a job. That's the first thing I want to point to you. The second thing is that in the theoretical world, if you imagine you're starting from a single copy, in the end, you should have this exponential growth that should follow 2 to the power of the number of cycles. So, if you do 2 cycles, you would have 2 to the power of 2, 4 copies, 3, 2 to the power of 3, 8 copies, and so on. So you could actually predict in the theoretical world how much you would get. This, however, is not really true, even though we like to teach you that. The growth of the samples in your PCR follows a curve similar to the one you see here in yellow. And this follows ultimately this formula right here, where you have that the final number of copy, x, after n number of cycles, will be equal to the number of copies you had at the beginning, so x 0, okay, that's the starting number of copies that you have of targets at the beginning, multiply by 1 plus the efficiency factor, with this sum being to the power of the number of cycle n. Now, it looks scary, it's actually very simple. First of all, the efficiency here, it's what really dictates how much you get in here. The efficiency can be anything between 0, meaning your PCR is not working, okay, so there's a problem with your primers, whatever you forgot, one of the deoxyribonucleotides, or to 1, where you have maximum 100% efficiency. Now, in the theoretical scenario that we were pointing to here, you will be with an efficiency of 1. Okay, so in this case, so if you have maximum efficiency, so this is 1, this will become 2, right? So if you're starting from 1 copy, this becomes 2. In the end, yes, you would have that growth that is proportional to 2 to the power of the number of cycles. However, this is not true. Efficiency is not always 100%. Actually, rarely is 100%. Basically, never. You would always have the sum of these copies, imagine later on, for some reason don't anneal properly to the primers. Maybe even 1 in 1,000, but that reduces efficiency. And what you have also is that this efficiency varies through the PCR. Is much usually higher at the beginning, but then later on gets lower and lower. And you see particularly here in this plateau phase. Before we get there, let's look at how this look appears through the different cycles. So here in the x-axis, you have the number of cycles of PCR. So this will be 0, 1, 2, all the way to 34, 35, whatever you got. You set up. And this is the number of amplicon molecules produced, followed in this case by fluorescence. This is a picture taken from quantitative real-time PCR, but applies to regular PCR as well. So what you have is that at the beginning, obviously the number of copies is growing very slow. If you're starting from very few copies, it'll take time. It'll be 4, 8, 36, so not a many. But at a certain point, at a certain cycle, imagine this being cycle 10, you start having that this copy become a significant number to start having this massive exponential growth. This is the exponential phase of PCR. The first one is called the initiation phase. In the initiation phase, until you reach this point, the amount of DNA you have is basically undetectable. Just to give you an example, imagine you stop your PCR here at cycle 7, for example, and you take your sample, you load it into a gel. Most likely you won't see anything. You would not have enough DNA to actually make it visible in the gel to be detectable with red safe or ethidium bromide, whatever is the molecule used to visualize the DNA. Or even by fluorescence, you will not be able to see it. But at a certain point, you start having enough DNA that start becoming visible. You pass the threshold there. And this is when you have the exponential phase. Boom. Now these molecules are growing super fast. You start having a lot, and the reason being that here you still have plenty of primers, plenty of nucleotides. DNA polymerase has not started denaturing yet, until you start reaching a point here where you start running out of some of the reagent. Not enough primers anymore. They can't keep up with all the copies of DNA that you're making. Some deoxyribonucleotides are not working anymore, you don't have them enough anymore, and so on. And so here you start reaching a plateau phase right here. But as you can see here, the efficiency too is changing through this. So E is not always a fixed value. It's probably constant through here, and then here start going lower and lower up to reaching a point of basically 0 when it's completely flat will be equal to 0. Okay. Now, a few words about DNA polymerase. As I say before, DNA polymerase can actually at a certain point be denatured. This was a huge problem, particularly at the very beginning in the 80s when the technique was just developed. What scientists were actually using was a truncated form of DNA polymerase 1. If you remember from DNA replication, DNA polymerase 1 in bacteria, so E. coli, for example, is that DNA polymerase that is involved in removing the RNA primers and replacing them with DNA. So it has 3 subunits actually present in there. It has a subunits here, so a domain. Now you should know, we talked about proteins in biochemistry. So it has a domain that is involved, has the enzymatic activity involved in the removal, the exonuclease activity to remove the ribonucleotide, so the RNA in the DNA. And then it has another domain, which is the one that finally starts synthesizing, attaching the deoxyribonucleotides, forming DNA. And together with that, it has a third domain that actually controls the accuracy of the DNA synthesis done by this domain. So it's referred to as the proofreading domain. The cleano fragment, which the one that was using from the very beginning, misses this primer removal domain. Okay, so it will be this one, the cleano fragment. In fact, you don't need it. There are no RNA primers to be removed, so all you need is these 2 fragments. The problem with these DNA polymerase, though, is that being isolated from bacteria, like E. coli, it's very sensitive to high temperature. It works well at 37 degrees, but then if the temperature go to 95, as you have in any denaturation step, not only that, but also all the other steps, annealing and elongation are done at high temperature, it basically denatures, this protein denatures at every cycle. So in fact, at the beginning, it had to be replaced. In every cycle, they had to add more cleano fragment to keep going and going and going. So imagine you have to do it for 30 times, 35 times, so depending on the number of cycles you're doing there. Then later on, a new polymerase was discovered from these bacteria, Thermus aquaticus. This is a bacteria that works at or survives at very high temperature. The reason that it survives is because it has enzymes that are much less sensitive to denaturation. In fact, DNA polymerase has an optimal activity in the range of 70 something degrees, 72, 74 degrees Celsius. Okay, so not only this polymerase can work well at high temperature, but also it does not denature much. Okay, so these have become the gold standard. So you did not need to replace it in every single cycle. Now there are modified versions of Taq DNA polymers done by different companies with their own names used. And there are also other DNA polymers discovered from other organisms. The main one is usually Pyrococcus, is a different type of bacteria, which is thermostable as well. The common new DNA polymers used now is PFU, which is from Pyrococcus furius. That's where the name comes from, PFU DNA polymers, which similar to Taq DNA polymers is resistant at high temperature, but it has an advantage, for example, compared to the Taq DNA polymers, is that it has a 5 times less error rate. So Taq DNA polymers basically introduces a wrong nucleotides every 9 to 10,000 nucleotides. The PFU will be 1 every 50,000. So if you wanna, let's say, amplify a sequence of DNA so that then you want to clone it to express a gene and so on, then you most likely use a PFU, not a Taq DNA polymers. Now let's focus on the primers. So let's go through a few roles that are used in the design of the primers particularly. Okay, so the primers, first of all, are these star of PCR. These are the sequence of DNA that do replace They have that dual function. They replace the RNA primers needed in DNA polymerization, in DNA replication, and most importantly, now they direct the DNA polymers to copy exactly the sequence that you want to copy. Okay, what you're interested in, the target sequence. The reason being that these primers are designed to bind exactly where the target sequence is, right? So that's the one that point to the target sequence and therefore cause the DNA polymers to amplify exclusively that sequence referred to as the amplicon. So they dictate also the size ultimately of the amplicon, which will go from the beginning of this primer to the end of this one. When you read about primers, oftentimes you see them referred as left or forward and right or reverse primer. And usually by definition, the left primer will be the one. So if you write your target DNA with a 5 to 3 prime strand on the top and 3 prime to 5 prime strand to the bottom here, the left or forward primer will be the one binding upstream, if you want, your target sequence here on the 3 prime to 5 prime strand. So this will be your left or forward primer, and the other one will be the one binding to the 5 prime to 3 prime. So at the end of your target sequence will be the one 3 prime to 5 prime here. Obviously they could be switched if these two sequences are written in the opposite order there. And one important thing is that always the direction of these primers, obviously this one has to be pointing DNA polymerase so that it will go over the target sequence. The same thing for this one. If you actually, this one was designed to bind in this part of this strand, right? Now the DNA polymers will go away from the target sequence. The same thing if this primer was designed to bind to this sequence right here. It will lead to the formation. So be extremely careful, important thing to do when you design your left and right primer right here. Now, these primers are designed, now there is a lot of online tools that make your job much easier. So if you know your target sequence, right, you can just extract it from a database like NCBI, extract it from there, enter it in these online tools, and then they will give you a series of set of primers that you could use to amplify that sequence. You would have to enter some criteria like how big you want your amplicon to be and so on. But you can actually design them even by hand, this primer, by following some specific rules and standards. So what is important when it comes to design primers is, first of all, the length of the primers. We'll discuss all of these in separate slides from starting from the next one. Then you always have to check that in their sequence these primers do not interact with themselves or with each other. They don't form secondary structure or anneal with each other. Then you have to follow specific GNC content rules. Okay, and then you want to make them so when it comes to actually do the experiment, you want to use them a specific annealing temperature, which is dependent to what is called the melting temperature. We'll talk about that again in a few slides. Other important thing is that when you design two primers, obviously, since they are in the same reaction, right, they go through the same cycle, the same temperature, these primers should be matched in terms of properties, particularly when it comes to the annealing temperature right here. So let's start with the primer length. Primers usually, again, you can find different numbers. Usually they're between 18 to 24 nucleotides long. You can go a little bit lower. It depends also on the genome you're working with and so on, the target sequence you get and so on. But let's take as a rule in general that they have to be between 18 to 24 nucleotides long. Now the problem with going with primers that are smaller than 18 or even much smaller than that is obviously the chance of them binding to sequences which are non-specific. Obviously you want them to bind to the target sequence, but the smaller you go, the higher is the chance that they might find a complementary sequence somewhere else in the genome, right? Their chance is proportional to the overall length. So just to give you a crazy example, imagine you have a primer that is 4 nucleotide long. The chance of binding to a sequence just by chance anywhere in the genome will be 1 over 4 to the power of 4, the number of nucleotides, right? So that will be a very small number. So actually that primer, 4 nucleotide long primer, will bind to many unwanted sequence. So the higher is the number of nucleotides, the smaller this chance becomes. You can do a calculation there. What will be your chance with a 17 nucleotide primer? Will be 1 in 4 over a power of 17, right? For example, if you're using this in a genome, which is 3 billion copies, that chance start becoming fairly high, right? So incompatible sometimes with a good experiment or to get good results. So you want to be bigger, going bigger than 18 nucleotides, but not too big. 24, 25 nucleotides is the maximum you can really use. The reason being that if you go longer, now the longer your primer is, the longer it will take to actually anneal to the target sequence, so form the double strand with the target sequence. And that would actually slow down your experiment significantly. It will take us so longer to be separated later on in every step. Now, another important feature that you want to keep in mind when you design your primers is their GC content. Obviously, the more GCs you have in your sequence, the stronger is the bond of the primers with its target sequence. But you don't want it to have it too strong, the bond, right? You want to have it strong enough so that it easily binds during the annealing time, but then you can separate it later on. So the rule of thumb is should be between 40 to 60%. 45-50% will be the ideal target. Other important feature that you should always look for is to have 2 or 3 GNCs at the very 3' end of the primer. So, for example, look at these primers right here. So these are 3 primers. Let's assume that they all would work well for what you're trying to do, right? Which one will be the ideal one? Now, you will look again for 2 things. Which one has a GC content between 40 and 60%? And you can see clearly this one, for example, is already 70 or 75. I remember I can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13 out of 20 nucleotides, right? We are at 65, actually, sorry, percent here of GCs. This would not work very well, even though it has 2 C and G at the 3' end here. So, but still would not work, would not be ideal. This one has less GNC, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. This is a 50%, has 2 GCs at the 3' will be a great primer to use. What about this one right here? Well, the GC content looks just at a glance okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it's 50% GNCs. Looks okay. But if you look, compared to this one, it doesn't have the 2 C and G at the end. So these will form, probably be the best primer, will form a good, fairly tight bond at the 3', which is where the end, where the Taq DNA polymers should start working. So this end here will be much more stable than this one right here, giving a better start for Taq DNA polymers. So this primer will be probably the ideal one to use. Then until you do PCR, you never know. But in theory will be the ideal one. Other important features when it comes to design the sequence of a primer is you have to look for sequences within the same primers or between the 2 primers which are matched with each other that can form hairpin. Like in this case, look at this primer right here. If you look carefully, I highlighted it here, much easier once you see it in bold, but sometimes you don't notice this. But if you look at this sequence right here, so if you read this sequence in this direction, right, and this in the opposite direction here, you see that there is complementarity here. So this, just to use the pen here, just to point it to you, this nucleotide will match with this one, this with this, this with this, and I guess you got the pattern by now. So basically this primer will tend to fold on itself, right, forming what we call a hairpin. We talked about that in biochemistry. And this obviously would be a primer that would not work. If it folds on its own, obviously it would not anneal to the target sequence. Now you can have a problem also with primers that they can have sequences that allows them to self-anneal. So the same primer can actually form a dimer like this one, or the two primers like in this case right here. You have that this T, again going back here with the pen, let's use maybe red, better. These would anneal with this with this with this, and so on. And so basically will lead to these two primers to anneal with each other, which then you would have competition between the cross annealing of these two primers and their annealing with the target sequence. This would greatly lower the efficiency of your PCR. Other things, very important thing that you have to keep in mind when you design primers is their melting temperature and their annealing temperature, which is related to the melting temperatures. So by melting temperature, what we mean is that temperature at which half of the primers, 50% of one of the primers, is dissociated from the DNA, or the other 50% will be associated to the DNA. So you have half which is annealed to the target DNA. Okay, could be 58, 60 degrees, 72 degrees, whatever it is. Okay, obviously this is proportionate to the amount of G and Cs you have. The higher the content, the higher is this temperature. The lower is this temperature, sorry. And the annealing temperature is that temperature that you use in the PCR to get the primers to bind to the target sequence. As a rule of thumb now, once you have determined the melting temperature of your primers, and I show you in the next slide how you can grossly do it, you would choose a temperature, an annealing temperature in your second step in your PCR cycle, so in the annealing step, which is 5 degrees lower than the predicted melting temperature. Okay. Now, if you use a temperature which is higher than the annealing temperatures, lower than the annealing temperature, now this primer would tend to bind to other sequences, right? Not maybe with the full primer sequence, but with some part of it, could still form a specific, non-specific binding. If you go to temperatures that are much higher than that, then at this point the primer would have a hard time actually annealing with the target sequence too. So you would not anneal, you would not have any formation of PCR product, or very little. So how can you predict the melting temperature of a primer? As I said before, it's proportional to the content of G's and C's in the primer itself, which should be between 40 and 60%. And roughly you can estimate it by adding 4 Celsius degrees for each G's and C's that you have in there, and 2 degrees for any T or A that you have in there. So for example, in this primer right here, what would be the temperature? Well, let's calculate it. So the temperature here, you would have, how many A and T's do you have in here? 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10, and 11. So you have 11 A's and T's, so you multiply by 2 degrees Celsius, right? And then how many G and C's do we have? 1, 2, 3, 4, 5, 6, 7, 8, and 9. It's a 20 nucleotide primer, so 9 times 4 degrees Celsius, right? So we have 22 degrees Celsius plus 9, that's 36. This primer, you could estimate it would have a melting temperature of roughly 58 degrees Celsius. So what would be the ideal annealing temperature to use? This will be this minus 5, around 53 degrees Celsius. Okay. Now in reality there are other factors that do determine the melting temperature and the annealing temperature. Other than this is a rough way to estimate it. In fact, if you take this primer online, there are some tools that allow you to predict it. You can get different values, and depends on the concentration of the primers, the type of DNA polymers that you use, and other factors as a concentration of salt and so on. So there are different things that might affect the melting temperature. But this is an easier way, a fairly easy way to get a start, okay, to estimate the melting temperature of any primer that you might have designed, and therefore the annealing temperature that you should use in your PCR. Now, let's go through an example. So we talked about the PCR. We talked about how it works, how the growth is, which is now really at the 2 to the power of the number of cycles. We talked about the role of primers, how you design them, what are the important factors and parameters that you have to keep in mind when you design the primers. Now, let's talk about how the application of PCR. PCR is used for many applications. Okay, I've used it for many in my own career, but I'm sure there are many more other applications than the one I used it for. So you can use it. It will go through an example to analyze genomic DNAs to do forensic studies. We'll go through the next slide about some example here. Or you can use it to clone a desired target sequence. Let's say you're interested in cloning a specific gene or even a promoter you want to study. That's how I've done it. For example, wanted to study how the Duchenne muscular dystrophy promoter works, how is activated in muscle cells or neurons and so on. You can actually clone that promoter by doing a PCR that goes through, right, the promoter. So if you design a primer before and one after the promoter, right, then you can make millions of copies of that. Then you can include that into a plasmid, and then you can do different studies with that. You can do that with a gene as well, the coding sequence as well. For example, you can that's how insulin can be cloned. Obviously though, in this case, if you want to do some cloning, you want to make sure that what you amplify has no mutations, right? As we said before, there is an error rate of the DNA polymerase. So once you do your PCR product and you clone it, then you would have to sequence that sequence just to make sure that it corresponds 100% the same than the original sequence. Now it can be used also to detect the presence of pathogens in biological samples. For example, in the lab that you'll be doing a PCR on this gene called the UIDA gene from E. coli. That's a technique that is commonly used to detect the presence of E. coli in sample, for example in meat. If you're doing meat testings in a facility where they process meat and so on, you want to know if there is contamination from E. coli. You would take a sample, obviously you would extract, you would get some buffer and so on to extract it from the meat and so on, and then you could test the presence of genomic DNA from E. coli by using PCR. This is an extremely sensitive test. Okay, so even if you have very little E. coli there, you will be able to detect it by PCR. So there are several applications where PCR can be used. Now I go just through one example which I had already mentioned in biochemistry very quickly, but I'll expand a little bit here, is how PCR can be used in forensic studies. It's commonly used in scenarios where, for example, there has been a murder. You want to know who potentially, you have a series of potential person that might have done that murder, and the first thing you would do, as you know, you would look for biological samples in the murder scene. You will look for a hair, you get the hair follicle, try to extract the DNA from the cigarette butts, and any possible evidence you would get from the whatever weapons was used. You can try to get some biological samples there, remain of skin left or underneath the nails of the victim, usually fight back, right? You extract the DNA, and then you could study that DNA by PCR and try to see if it matches any possible suspect in that murder, right? How do you do that? You do that by studying some specific loci, which are called variable number of tandem repeats. Before we talked about variable number of tandem repeats, let's talk about loci. Loci is the plural of locus. Locus means any sequence in the genome that is conserved. Okay. Oftentimes, we talked about loci or locus when we talk about coding genes, right? Obviously, if this is the sequence in this chromosome, I say that codes for a specific protein, this will be a coding gene, right? Common thing exists in different alleles, one from the mother, one from the father, and so on. But there are other sequences that do not code for genes. They are still conserved, right? And these sequences are preserved as well as the coding genes, and they can exist in different forms. but this still thing, this sequence is present in any chromosome. I take my DNA, your DNA, you will still find this sequence conserved. Might have some variation, again, different alleles as well as coding genes, even though it's not non-coding genes. All of these are referred to as loci. Now, variable number of tandem repeats, or VNTRs, are some of these non-coding sequences, which, as the name said, instead of being made by coding sequences they're made by some sequences which are repeat several times. For example, look at this sequence here. I just made it up, okay? This sequence here is exactly like this one, like this one, like this one. This is a sequence which is repeated in tandem, right? Several times, in this case 5 times. This could be a locus, a variable number of tandem repeat type of locus, okay? And you have many types. In our genome we have several of them, all right? And they're conserved. They're in different chromosomes. They would have different sequences, okay, that are repeated. They're not all the same. For example, D1S80 has a 16 nucleotide sequence, so something a little bit longer than this, repeated n times. But again, they are conserved. They must have some function in our genome since they are conserved through evolution. They don't get lost. But the other important thing is that now, when you look at these VNTRs, now you can have different versions or different number of these repeats in different individuals. So, for example, let's say this one has 5 repeats, another version might have 7 repeats, even though the sequence is the same, but it's repeated more than 5 times, 7 times. This difference, these 2 different versions, are referred to as alleles, exactly like in coding genes. So an individual may have 5 and 7 repeats. Another individual might have 11 and 14 repeats. Okay. Now, because this number of repeats are different between different individuals, and obviously you can have 2 if you're heterozygous or 1 if you're homozygous, now determining this number of repeats can greatly help to determine the actual identity of a DNA in a forensic study. Now, a very common VNTR that is used is what is called the D1S80. This is actually a VNTR that oftentimes we use in a lab at Seneca, but unfortunately now, since we have to take some biological sample, usually from saliva, in COVID times it cannot be done. But it's a really, so you would actually take your own genomic DNA, you would extract your own genomic DNA and test yourself for this VNTR. Unfortunately we can't do it, but still, great example of a VNTR. So this VNTR, D1S80, is located in chromosome 1, all right, at the very tip here, the very end here of chromosome 1 right here. So it's conserved. You have it there, I have it there. Each one of us has 2 copies, one coming from the father, one from the mother, obviously having 2 copies of chromosome 1s. Now, this VNTR is made by a repeat, like this one, which is, however, 16 base pairs long. And now, different individuals, so different alleles here, can vary in the number of these repeats that you may have. You can have anything between, in your alleles, anything between 14 to 41 different type of repeats. There are 29 of them possible. There is some math problem there. Obviously it's not 29, but it will be 27. Now, most individuals would have 2 different versions here. So about 4 out of 5, 83%, will have a certain number of repeats in one allele and a different number here. So they will be heterozygous for that. But 1 in 5, about 20%, 17%, would actually be homozygous. We have the same number of repeats there. Now, you can actually visualize the different number of repeats in the D1S80 and therefore fingerprint the DNA of an individual by using PCR. Not only you can visualize it, but also determine the number of repeats that you have in there by PCR. So how do you do that? Well, this is done by using primers like this one right here, which are designed to bind right beside the D1S80 locus. Right? So these will bind beside, and so if you do the PCR here, now your amplicone will go from the beginning of the left primer to the end of the right primer here, right? So it will be this size here. And the size that you would get out of the PCR obviously will be different if an individual like this one right here is heterozygous, would have one allele with a certain number of repeats, and then the other one from the mother in this example with less repeats. This will give you an amplicon which is bigger, right, the paternal one, compared to the one coming from the mother, maternal one. The same thing for this individual. Here you can see this individual here instead seems to have two alleles identical to each other or with minimal difference. Keep in mind that this is an agarose gel, doesn't have a excellent resolution, so at 16 base nucleotide repeat difference or base pairs repeat difference here might not be visible in this type of gel. So, but this individual here clearly has a different there. So each amplicon, this one or this one, will be coming from the different alleles, one from the father, one from the mother here. And now you can even determine the size. We'll talk about at the end of this lecture on how you can use ultimately a DNA ladder here to actually estimate the size of any DNA band, in this case these amplicons produced from the D1S80. So if you can figure out the size of these bands, now you can figure out also the number of repeats, because you know that the total size here will be coming from the beginning of this primer to the end of this primer. If you know where these primers are located compared to the beginning of the NTR, let's say you know that this from here to here is 42 base pairs, and here from here is 34, so this size will be 42 plus 34 plus 16 times the number of repeats. So if you can determine the size of here, subtract these two distances here, divided by 16, you will get the number of repeats that you have in there. So these to point to you how you can use PCR ultimately to determine the number of repeats present in different individuals. So you can see how in forensic studies, so now if you have some DNA samples, I don't have an example here, but imagine this sample 4 is the one that was extracted from some DNA underneath the nail of the victim. Okay, just by matching visually what you get in here compared to the one taken by these three suspects, in this example, you can see how in this case you could conclude that these suspects are not the one that own the DNA coming from this tissue here. So they're not most likely the people that have been fighting and murdered the victim. Okay, we're gonna take a break here, and then we're gonna resume in the second part talking about modified version of PCR.